You want to mute? You want to mute everybody too?
Yeah, hang on. Good point. Thank you. Thank you for reminding me. Share screen. Okay. Oh, hang on a sec. I need to go back. Yeah. Okay. I had to switch off my second screen. You end up with both screens. So some of you may have seen this picture before. It's an old painting, and it's a painting of a poor fellow who is just about to die, and he is being infected with tetanus. Now, the word tetanus comes from the word tetany, which means the contraction of muscles. And the toxin that the tetanus bacteria produce under the skin, they don't move around the body, they just grow in one spot and produce a substance which causes contraction. And the thing is, every skeletal muscle in the body will contract at the same time. So all the bones, one at a time, just snap. It's a terrible, terrible death, and a very, very painful one. So this guy, this picture actually shows what people would have looked like in the last a few minutes. So we're talking about pathogens, and there are different sorts. Some which are cellular, made of cells, and some which are not. And so the ones which are non-cellular, we have these prions. I'll go through all these in detail, and the viruses and the viroids. I should just mention the viroids only attack plants. So that's one less we have to worry about. And then the cellular agents, the bacteria and eukaryotes, and they're the multicellular organisms. Now, all of these are what we call antigens. They set up an immune response, except for the prions, and you'll see why very shortly. So we have this amazing system in us called the immune system, and it prevents the foreign. Generally, the idea is to prevent the foreign antigen from coming into the body. And once they're in the body, we have to try and remove them. And we also have to make chemicals, if you like, antibodies, for future protection of these antigens which have just hit us. So it's important to have a bit of a historical perspective to this. So in the 14th century, the Black Death killed huge numbers, many hundreds of thousands of people in Europe. And it wasn't until the 19th century that bacteria were identified as the cause of the Great Plague. And they showed that it was the fleas that were the vector, were the things that were moving the bacteria around. Now, fleas normally live on the rats, But if the rats are a little bit on the short supply side in some areas, they will jump to humans, and some of them may have been tried to suck blood out of an infected person or an infected rat, and then go to an uninfected human, and then they pass it on. Now, but far earlier on in the 14th century, they had no idea, and they didn't know what it was. They didn't know how to control it. They developed these masks. They closed all the doors and windows to keep the wind out. They thought it may be in the breeze. They attacked foreigners who they thought would bring them in, and different religions were massacred. And they just didn't know what was causing it. They even whipped each other because they thought it was God's punishment for something they did or didn't do. So it wasn't until the 1850s when Louis Pasteur actually found bacteria. He discovered that he watched them grow, multiply, and they and even though they knew about, a small number of people would have known about bacteria, there were many ideas, different ideas about diseases. Now, Louis Pasteur did not work out, or did not, I wasn't going to use the word discover, but didn't make a microscope, but he certainly did use them. Microscopes were done by quite a bit earlier, and one guy called Leeuwenhoek was very big on actually producing the new microscopes. Now, in China, in Europe, they had the miasma theory, and that it's been there for a long, long time. It just wouldn't go away. And it really wasn't until 1880s when good old Louis, his idea of pathogens was made more popular, came the germ theory, something that caused disease. Now, of course, once they knew about bacteria, didn't mean that they had any idea about viruses, which are many, many times smaller, and you can't see them anyway using a microscope. So the miasma was thought to be a poisonous vapor, some sort of a mist filled with particles from decomposing matter. And this decomposing matter is called miasmata, and that caused sicknesses. So the miasmatic position was a product of the environment. Things like water, bad air, poor hygienic conditions. And again, the idea of the germ theory, this is before the germ theory. So they had no idea. Now, under the miasma theory, excuse me, such infections wasn't passed between individuals. It was from the environment, and it is thought it's identifiable by a bad smell, foul smell. So it was propagated, they thought, through worms from ulcers within people affected by the plague. So they had no, again, it was all these amazing ideas. Now we have to be careful not to use the word theory. These weren't theories, but by definition, you have a theory when you have evidence. So these were, if you like, hypotheses or ideas. So they dressed up like that to try and keep everything out. Here I put a word, a flea, picture of a flea, and these are photos, very large enlargements, if you like, of the of bacteria causing the plague. So in 1665, during the plague, the doctors had this costume, and it was ankle length, it was made of canvas, and they had a beak like a bird, and they had this material which they soaked in some strong smelling sweet stuff like lavender maybe, and they had gloves, boots, wide brimmed leather hat. Why the hat, no idea, and an outer cloth covering garment. So the guy goes around. The stick, by the way, was for turning bodies over just to check. They didn't want to touch the body. So it had a glass opening for the eyes, a curved beak, and so they had holes in it so the doctor could breathe, and with the material inside. So here's another one, very similar, and the writing is in German. So if any of you have the German knowledge, I can't remember whether I put this in the notes or not. Now the beak, I did some further research, and what did they actually put in the beak? They had dried flowers, they had herbs, and some of them had some spongy material and soaked it in vinegar. So they had the vinegary smell. But don't forget, there were people dying in the streets. The place, there was no sewage either. So the smell would have been terrible. But they didn't know which of those smells was the miasma causing the problem. So the doctors had to keep these smells out, from that, stopping them from getting infected. Now the wide-brimmed hat indicated the profession. That's what it was, but it was no use. And and as I said, the wooden cane was to point to areas and examine patients without touching them. And it just to keep people away from the doctor. Again, they didn't know what was causing it. Now, smallpox. It's been around for a very, very long time. It now does not exist in the world. It does exist in very, very high security laboratories, because they may need to access the genetics or something sometime in the future. And apparently there's been, it's thought that it first appeared about 10,000 BC in northeastern Africa. So through prehistoric, there weren't documents, but stories or just the culture from the time. So in the 18th century, probably killed about 400,000 people each year. And it's exceptionally contagious. And with smallpox, variolation was attempted, and I'll get to that, by taking samples from smallpox vesicles. So little bubbles that form in the skin, has a little bit of liquid in it, and scratch the skin or the nostrils of a healthy person and introduce some of this liquid. And this was made famous in the 1790s by an English doctor, Edward Jenner, who noticed that cow maids, they're the young women who milked the cows, who developed cowpox, which is similar to smallpox, but they did not develop the smallpox. And so he got permission of some people, a couple with a young boy, to experiment on him. So he got some liquid from the hands of the cow maids, and he inoculated, if you like, the boy with this material from the cowpox, from the cow maids rather. Now, soon after that, he infected the boy with the smallpox, and the boy was immune. So this was the actual beginning. So we're talking about 1790s. I couldn't find an actual date. And so this started the whole idea of immunization. And you should know that vaccination comes from the Latin vacca for cow, meaning the cowpox. So then it led to the next thing with rabies. When people were bitten by rabid dogs, it didn't take long for them to die a really nasty death, a bit like tetanus. But it's a viral disease, and it inflames the brain. So that causes problems. Even in Australia, we used to have rabid dogs. Dogs carried it. And if you saw a dog with foaming saliva around the mouth, the dog was very quickly removed and destroyed. And before Pasteur discovered the cause of rabies, well, he knew it wasn't bacteria, it was virus, but he couldn't see it. But before he got involved, they used to cauterize the bite of the dog with a hot iron to try and kill whatever was caused, but it wasn't particularly useful, and people were burnt badly. It wasn't much useful. Now, Semmelweis is a very, very famous Hungarian doctor, and he just in 1847 he insisted that the doctors at the hospital where he worked in Vienna washed their hands. Working with it's calcium chloride. It's chlorinated lime. It's called. Now, what happened was doctors, obstetricians delivered the babies, but a lot of the women died and they were taken to the morgue. The doctor went into the morgue to see what caused the death of the woman. And then there was another delivery, so he went back. And in doing so, he was moving the bacteria that caused the death of the women in the morgue. He passed it on to the next woman who was giving birth. And the condition was called puerperal fever or sepsis fever. And sepsis is where you become infected. And it was also called childbirth fever. A large number of women died. And Florence Nightingale noticed during the war that soldiers in hospital beds who were next to each other, but they weren't wounded, they may be wounded, but they had other infections, were passing on the infection, whether if they were separated with a greater distance, they weren't. This sort of idea of doctors noticing or nurses noticing things made a change. And he noticed that in hospitals where doctors didn't actually do the obstetricians didn't do the post-mortems, they didn't have puerperal fever. So they knew about lime solution. I don't know the history of that, but he insisted that doctors, when they come out of from post-mortem, they wash their hands, and as soon as that happened, not a single woman after that died of puerperal fever. And that when eventually there was a lot of resistance from hospitals and doctors around the world, they eventually moved on. Malaria, it comes from the old Italian malaria, bad air, because they thought that it came from the bad smells are from swamps. Now, it used to be called marsh fever. They always thought that the swamps and the bad smell, and people died. And the association, well, swamps usually have mosquitoes. But the thing is, you get stuck by a mosquito, but nothing happens for a while. Now, how often have we been stung by a mosquito and within a few minutes, unless you scratch it, of course, we forget about it, and there's no link. So they call it marsh fever. But eventually that was discovered too. The Spanish flu, during the war and post-war, First World War, killed between 20 and 40 million people. Australia was immune completely until after the war, when the soldiers were coming back home from Europe, and they brought it with them, and then it spread, killed thousands of Aussies. Today, we get between a quarter to 500,000 people who die annually of the flu, and most of them are elderly. Now, not everyone gets immunized, and this morning there was a doctor on ABC who was saying we had very little flu in 2020, a lot less in 21, and they expect that this year might be a bit of a bonza year for flu, because people are forgetting to go and get immunized. I've had it, and it's been fun. But we still have a lot of people dying in Australia from flu every year.
Ruben, can I ask you something?
Sure.
It's a question I have. How much flu was there in other parts of the world in
Good question.
in their winter?
Yeah.
And if there wasn't much because of the pandemic, how did they know which strains to put into the current vaccine?
A brilliant question. I haven't heard. I don't know. I haven't seen, and I don't know which four they picked. So no idea. You're right. They normally look at what's common in Europe and Asia. I'll have to do some research on that. I will, if I find out, I'll send it out to you. Now, these numbers here, you see H1N1, H5N1, H7N1, they actually refer to the flu virus, because there are different forms of it. Now, the H stands for hemagglutinin, and the N for neuraminidase. And so again, I'm giving you two examples. In wild birds, because wild birds spread flus around, there are 15 forms of the hemagglutinin and 9 forms of the neuraminidase. In us, it's 18 and 11. So H1N1 would be these ones, and they would infect us. And well, you know what the flu's like. And then, but they do modify. The hemagglutinin helps escape. It comes later, but it's just to make sure you have an idea. The hemagglutinin on the surface of the virus helps escape the cells that produce them. But they're still stuck to the cell. The neuraminidase, ASE, means it's an enzyme, cuts the hemagglutinin chemically, and then it's released from the cell that produced it, and then it goes on to other cells and to infect them. But there are also the variations, like this Chinese one here, H7N9. You may see them in the papers as well, listed as with these numbers, just so an idea that you have an idea where that comes from. Okay, so I'll get back to that later. Now, we have two types of immune systems. We have the non-specific immune system and the specific immune system. The non-specific, we have two lines of defense. And the specific, we have a third line. Okay, so this is going to be, as I said, this is a fairly dense talk. The first line of defense that we have is what you see on the outside. We have skin and we have a keratin layer. Now, keratin is tough. All our skin has got this protein keratin. Our hair is keratin, our nails are keratin, horses' hooves are keratin. If you open us up and look at our livers, kidneys, they're quite soft. They don't have keratin. It's only the outside, and it's a protective layer. We have mucous membranes. So in the nose, the mouth, the vagina, the rectum, they all have mucus, and that's also a layer of protection to prevent entry into the body, that is. We have natural secretions. We have saliva. We have acid in the stomach. We have tears. We have mucus. Now, the saliva has enzymes in it, as do the tears. The mucus layer is a layer. The acid, well, that speaks for itself. And we also have natural flora around the body. Flora as in flowers, but that just means plant material, plant type. And so things like bacteria and fungi. Bit more detail later. Now, once some pathogen gets into us, we have a second line of defense, but this is non-specific. So we have phagocytes. Now, phagocytes, phago is to eat, cyte is cell. And so these are white blood cells, the non-specific, anything that's not part of us is nasty, and they try and get rid of it by consuming it. We have natural killer cells, and they will kill cells that have been attacked. And a cell that is attacked by, say, a virus sends out a chemical signal, and the NK cells follow the signal and they destroy the cells before the viruses can multiply. So with the also called cytotoxic lymphocytes. So the word cyto means cell, toxic, so it's toxic to the cells, and they're the lymphocytes, the sites, the cells that are produced in the lymphatic system. So that's what these guys are, but they're non-specific. So we have complement proteins, and these are proteins that are constantly floating in our blood, and anything unusual, and I don't know what the mechanism is which recognizes self and non-self, what's unusual and what's not. But for example, if you get a splinter and you pull it out, some stays in there, it will become pussy. Now the pus is due to dead, I'll come back to that, dead white blood cells. But the first thing that happens, the splinter gets covered by these proteins, and these phagocytes here follow these proteins, and then they try to destroy whatever the proteins covered. Now they can't destroy the splinter, and they die in the process. So and that's where the pus comes from. Now we also have an interferon, which is a chemical. It's like a hormone produced by a cell that's been attacked by a virus. It sends out two chemicals, the interferon and the cytokines. The interferon is a signal to its neighboring cells, so the cells near this cell that's been attacked, to say, I have been attacked, do something for yourself, protect yourselves. So those cells that have received this information change the configuration of the cell membrane a bit to make it harder for the virus to get in. The cytokines are pumped into the blood system, and the phagocytes then follow the concentration of the cytokines to, well, I mentioned that before, but the cytokines are what attracts the white blood cells, the phagocytes, to destroy them. And they're also, usually the natural killer cells are there even faster. So there are two sorts. Oh, sorry, no, I made a mistake. It's not the phagocytes, it's the natural killer cells that are attracted by the cytokines, and that's why they're cytotoxic. They kill the infected cells. Then there's also inflammation. Now, inflammation is useful because the mast cells in our skin, usually, they produce histamine. Now, if you get scratched by a cat or prickled by a rose or something, you'll get a little red mark and it's itchy. The itchiness is due to the histamine that's being released by the mast cells because it's done to protect that particular area. And what happens is the histamines increase the blood, cause the blood flow to increase to that area, and that's why it becomes a bit redder, a bit warmer, and it allows the white blood cells to come out to protect whatever caused the problem. And of course, blood clotting is a very important aspect of protection, because if your skin is affected by a cut or something, the last thing you want is for the cut not to be sealed. So that's where the blood clotting comes from. It's important. This is a very good diagram which shows what happens. Here's a wound and bacteria coming in. The mast cell here are the mast cells release the histamines. They affect the I can't think of the word, the way that cells can, like these white blood cells here, there are two different sorts here, they can come through much more easily and get rid of the bacteria. The blood platelets come through and also start the clotting process. So the adrenaline does that. All this is red due to the adrenaline, and that increases the blood flow. Now the cytokines, this is a macrophage. It's a we're being attacked. I'm gobbling up, if you like, the attacker. They send out cytokines, goes to the blood cells, and then a lot more will come to support them. All right, so I'm sure I've added this diagram in. Follow it. It's really, really good. It's very, it's a very good representation of what happens with a wound. But again, it's non-specific, right? That's internal. Now, the third line of defense is the specific one. This is the one you hear about when we talk about COVID. So we have two groups of cells, the B cells and the T cells. The B cells are called the humoral response cells. Now, humor in Latin means liquid, and the B cells produce the antibodies. Now, the B cell, they when there's an infection, they get converted to what we call plasma cells. They produce the antibodies, and we also call them, these antibodies, immunoglobulins. Now, globulin means a protein, and the immuno refers to the immunology. So they're very, very specific to whatever is attacking us. It also, there's another set of the two types of B cells. The plasma cells, one, the other one's memory B cells, which just proliferate when we get attacked, but they have to be there. They wane away a bit, but they're there for the future if we're attacked by the same, whatever it is that attacked us. But the antibodies are moved around in the blood, and that's why the blood, as we know, is a liquid, and that's why it's called a humoral response. On the other hand, the T cells are called, their response is cell mediated, because they actually touch each other. They pass on a message through touch, and there are three of them. Now, the helper T cells, I'll put these in case you're doing some more research for yourself. They're called CD4+. I will not go there, it's just too complex. They're also called TH cells, T helper cells. Cytotoxic, the CD8s, and the TC cells. And then we'll also have memory T cells. Okay, this is where it gets a bit complicated. And these work together with the B cells and T cells work together. Sometimes, well, the B cells and the T cells actually need each other, and you'll see. Now, we talk about antigens, we talk about antibodies. The difference, the antigen is what causes a response. It could be a protein, a toxin of some sort, it could be cellular and non-cellular, but the body reacts and it produces antibodies to the antigen. Now, the exception are these non-living proteins called prions. I should mention that prions, it comes from infective proteins, and we all have, I'm trying to remember whether I've got it further down or not. I can't remember. I just mentioned it now. We have lots of prions, very large number of prions in our brains. And we know they're there, but the only thing that's come up in recent times that possibly could be there for is our memory. It's part of the memory process. We're not sure. However, there are two forms of it. In our brain, we have the good form. Sometimes a bad form forms. Yes, sorry, I just remembered. I just pictured the diagram, and I've got the diagram later. I'll come back to it. It's easier. So just going back to the B cells and the T cells, we have the idea of self and non-self. So on all membranes, all cell membranes, except for the red blood cells, because they don't have a nucleus, all of our other cells, we have glycoproteins. Now, glyco means sugar and protein. So these are proteins with a bit of sugar on them. And they're called MHCs. I'll get to that in a minute. And they are unique. And that is what makes us us. Now, it's where, for example, if one of you give me some of your blood, and no, not blood, say that tissue, my system would reject it because it's not myself. It's non-self. So these MHCs, I'll explain that in a sec, allow the body's immune system to distinguish between self and non-self. It's a complex system, and it works amazingly well because we're all here. If it didn't work, we would not be here. Now, these are two proteins, and on the top, they've got these little bits of sugar. And they stand for major histocompatibility complex, MHC protein, because it's a big word that we just normally refer to as MHC. Now, it's not that it's a major protein. The MHC refers to the DNA where these are the genes for these are kept, and it's actually a major complex in that section of the DNA. And that's all I'm saying. It's too much. So these MHCs occur on all membranes except red blood cells, and there are two. There's a class 1 and a class 2, and if every cell, except the red blood cells, have the class 1. So on all nucleated cells, the class 2 are on the immune response cells, the B cells and the T cells and some of the macrophages. So because these immune response cells, the B cells and the T cells, go around sampling all day. That's all they do, they sample. And they go around within tissues, within everywhere, sampling self. The thing is, they themselves get sampled. Now, so there must be some way of distinguishing ways of recognition with these different sorts of cells. So the class 1 markers on all the cells, including the immune response cells, but only these T cells and B cells have the class 2, and the class 2 markers sample the class 1. Okay, so they use that for their recognition. So these specifically recognize self. That's very, very important. And there are people when they're born that have problems with these class 2s, and they can get destroyed from within. They're not, the immune system just doesn't recognize them. They have also, well, that can help them now a bit. So what they look like is something like this, a protein sticking into the cell membrane there. There's a bilipid layer, so each ball would be, and the two, well, I'll leave it for another talk. So the two layers here, these are within them, and these are the sugars, so the hexagonal bits here. All right, so just to give you an idea of on the surfaces multiple times on every cell. Now, a non-self, that could be a protein, that could be a toxin or something, which is an antigen, and something that causes a response. That something that has not been produced by the body, they recognize as non-self, and these would probably belong to a pathogen. So we have the immune response. It's a series of reactions which keeps us safe and which destroys whatever has invaded and destroys and removes the rubbish that's left over from the destruction. Now, they also, if you get, say, tissue I mentioned before from somebody else, it's blood, and the blood type, the A positive, negative, AB, AB, and O, so it comes into it as well. We also have a system where all this is sort of regulated to some degree, the lymphatic system. Now, when we draw the blood circulatory system, we use red for arterial blood and blue for venous blood, and we use green when we talk about the lymphatic system. So it's an open system as opposed to the blood system, which is a closed system. Now, closed system finishes where it starts. It starts at the heart, goes on in the body, comes back to the heart. The lymphatic system is an open system because it starts in the tissues. And it works its way back to the heart and becomes part of it again. Okay, so the lymph is the liquid in the lymphatic system, is right through us. There are lymph nodes. Now you'll see in joints, especially, you see little dots here in the groin, in the elbows, in the armpits, in the neck. And they are the nodes through which the lymph moves. And they are very complex inside. They are filters. And within the filters, there are the lymphocytes. And if there's anything that, say, you scratch yourself in the leg and some bacteria get in, those bacteria or something else, it'll get caught in the tissue in the groin, or sometimes behind the knee, and they'll swell. When they're swollen, it means that the defense system is working on removing it. So these, if you like, filters are very, very important. If say you have scratched yourself in the leg, and you go to the doctor, said, I'm in pain, the doctor might feel in the groin for the lymphatics to see if they're swollen. If it's an arm, feeling the armpit. In your tonsils, for example, they're the lymphoid tissue. The thoracic duct's got them as well. So they're all over the place. So everything is filtered before it gets to the blood and to the heart, I mean, and mixing with the blood. The antigens that have been filtered are then destroyed by the lymphocytes. Remember, these are the cells in the lymph. So let's go into a bit more detail. The B cells, which are, remember we talked about the humoral response, the liquid response. The antibodies are produced, and this is a repeat of what happened, I said before, and the 3 cells in the T cells. So the lymphocytes, and now they are all produced in the bone marrow. The B cells are also mature in the marrow, but the T cells they're called T cells because of the thymus gland, which is in the neck. That's where they mature. But the B cells are not called B cells because they mature in the bone. They're called because the first place they were discovered was in the bursa of birds, B for bursa. And so the birds produce their blood cells in the bursa, not in the bones, because the bones are, well, flight birds, the bones are hollow. So I put this diagram to show you. So here's the thymus, so they have their T cells produced there, and the bursa is at the back. It's called the bursa of Fabricius. And this is in chicken, but all birds are the same. Actually, I'm not sure about the ratites, the birds that don't fly, because their bones are solid, heavy. Now, what happens with the B cells? So every B cell has multiple copies of one kind of a surface receptor for a specific antigen. Now, there are millions of different antigens which could affect us. And we basically have some cells with each of those possible antigen receptors. It's incredible. So if here's something, here's a bit of antigen, and the B cell responds to it, it takes it in, breaks it up, it takes the antigen in, and part of that antigen is placed on the surface, which is recognized by a T cell. The T cell then produces a signal to the B cells. Sorry. And the B cells all around, the specific ones, start to proliferate into plasma cells and produce the antibodies. These Y-shaped things are the antibodies. That's their job. And so within the entire population of B cells, we have millions of different antigens. So there are very few B cells with the same antigen receptor. So when the B cell matches with a specific antigen, there are, let's say, two reactions. The plasma cells proliferate, we call it proliferation, and they produce antibodies. Now, these are monoclonal. You've heard of, and I'm sure you've heard of monoclonal antibodies. They are just one type of antibody. Because this particular group here would only be able to produce one type. Now, the memory B cells are also produced, not for now, but for the future. And so, so the production of antibodies is called, as I said before, the humoral immune response. Okay, because all these antibodies are then pumped into the blood, into the serum. Now, the antibodies themselves, as I said, are very, very specific. So here's a lovely diagram I found. The actual antibody is this Y-shaped thing. It's got what's called a heavy chain and a light chain, and the joint in here that doesn't show it here. But I'm showing this one. Here are 6 possible antigens. Of course, these are just diagrammatic. If you look at the binding site for the antigen, this is the only one that would fit. Okay, so that gets sent out. So this variable portion is specific for that one antibody, and the ones that are produced in large numbers from those B cells, from the serum cells, B serum cells. So here is another diagram, similar idea. They are joined here by sulfur bonds. They're called disulfide bonds. There are the 2 sulfurs in there, and that acts as a hinge. because the antigens aren't all the same size. They have a different shape, they'll also have a different size. So this bit here, which is this bit here, can move and along this hinge. So the light chain moves away from the heavy chain, and so that the antigen can fit in. Now there are also, doesn't show it here, there are two sets of sulfur bonds here to hold it together. So there are 4 proteins in this each antibody, and the variable portion is what's specific to the antigen that's occurred. Okay. Now, sorry, once the antigen is picked up here, the system will not let it go. So here we have some these cloud-like things here. It took me a while to find something that isn't too complex. So antibodies will attach to it, to whatever's on its surface. And so the antigen then can't do anything. And so they become disabled. So we produce the antibodies, and you can see here the sulfur, disulfur bonds, sulfide bonds, and that's what it would look like. And then the leukocytes, the white blood cells, will come and engulf the whole lot, including the antibodies, digest everything, and get rid of them. Now, but this is a very, very important graph. It shows what happens when we get either immunized or get an attack, a primary attack. And the response, this shows vaccination, it's not the needle here. But if you've been infected, it's the same thing. So you get, we get our COVID shot, or we get our flu shot, and we get a small response, but of antibodies. But the memory cells are there. They weren't there. They're a very small number of memory cells, but they get quite a few memory cells. And then after a bit of time, they wane off. Time moves on, and then if you're attacked again, the response is massive and much, much, much faster. This is called a secondary response. And the number of antibodies that are produced is massive compared to the initial response, the primary response. And this is why we get immunized. Now, there are 5 types of antibodies that we produce. This is complex. I won't dwell on it too much, but you should realize that not all antibodies are the same. Again, they're called immunology, or globulins of proteins. So the immunoglobulins, the Ig, so we've got the Ig, A, D, E, G, and N. And they all do something different. And so the IgA is found in milk, sweat, and saliva. So basically, we produce them, we pump them into our liquids that we produce, and if anything comes up, they're very, very, they're not that specific. And if a bacterium or a virus or something comes along, they will get rid of it. The IgDs, they are the surface of the B lymphocytes. Okay, and this is where the activation occurs when they contact the T cells, and then you get them this. The immunoglobulins are then produced, and then they circulate. Okay, so these are on the surface of the B cells. They don't actually hunt down something. These are the Ds. Okay, They control B lymphocyte activation. Remember that where the B cells and the T cells communicate, it's these guys, the IgDs. And so, yeah, so they say here, there may be a factor in the active elimination of B lymphocytes. If there's too many of them, there's a self-reactive thing. I found that, but I thought I'll chuck it in just for the hell of it. Now these control that's right. Sometimes the immune response goes crazy and produces far more antibodies than there should be. And too many B lymphocytes. So these control them, this IgD. Just reduce the numbers. So the IgE is found on mast cells in the skin, and basophils, it's another type of white blood cell. Mast cells are the ones that produce the so this is a mediator in allergic reactions. So the mast cells produce the histamines, and so when an antigen gets under the skin or into the skin, the IgE molecules are there, bound to a mast cell, and then that diagram I showed you what happens when there's a scratch or something. It's the IgE molecule that gets the whole system moving. Now, lysis is what's called a lytic process, LYTIC, and it also is involved in that or chemical degradation of parasites. Now, lysis means the breakdown. So when the bacteria, they are often just broken down in the system. Their membranes, their coating breaks apart. Parasites, they're protozoans, like amoeba, for example, we've got them. It's the IgE that knocks them out. The IgG is in blood and the lymph, and it can cross the placenta in pregnant women, allowing to protect the unborn fetus and the newborn. So they're already born with these antibodies, and they involve in phagocytosis. Now, phagocytosis is when the phagocytes, the white blood cells, actually surround something like bacteria, for example, something foreign, and engulf it and get rid of it. So phagocytosis in macrophages and neutrophils. So these are non-specific, but they also neutralize toxins, and they inactivate viruses and they kill bacteria. So these are they're non-specific, and they just go for it. They knock everybody out. IgG can also bind to natural killer cells, I mentioned earlier, and they are part of a process called antibody-dependent, cell-mediated cytotoxicity. Do you like that one? And which can kill or limit the effects of invading microbes. So the antibody-dependent, okay, that's that bit. Cell-mediated, which is that bit, killing off the cells that attack us. And the IgM is the immunoglobulin made during the typical response, the first responder. So it agglutinates. That's what we talked about before. These are the ones that clump invaders. And And they also are involved in the breakdown, the lysis, the breakdown of phagocytes, or bacteria, whatever it might be. Okay, so these are the IgMs. So antibody production is there for them. Now, the T cells, these are cell mediated. So, so macrophages, they engulf the pathogen. So here's a macrophage. It actually, this sort of movement that you're seeing here, where these bits of the macrophage are pushed out by macrophage, it's called amoeboid movement. Here's a little cartoon I found of a macrophage attacking a bacterium. Bacterium, of course, thinks that's it. So it actually surrounds it and forms a little vesicle, a little bubble, and then it pumps the antibodies in there to destroy them. It's much easier with a cartoon. But so they engulf the bacterium or whatever's attacking it. And then some of these surface proteins, or the antigens, are added to, once it's in there, they take it apart, and some of the surface proteins from what's this material, this pathogen, are removed and put on the surface of this white blood cell on their membrane, and then so they are, so these antigen they become what are called antigen presenting cells. Leukocytes is a general word for leuco is white in Latin, white cells, with the antigens are called APCs, antigen presenting cells, which then are picked up, the surface bits are picked up by the helper T cells. Remember, there are 3 types of T cells, and they recognize them as non-self, and then the signals go out for their destruction. So the helper T cells then secrete a protein called a cytokine. Now the cytokines, you may have seen that in that little table, had little yellow dots, yellow spots. They stimulate the cloning of B cells. Now the B cells, very specific, clone for that specific antigen from whatever's been attacking, and it's being gobbled up by these white blood cells. Okay, so if there's one or two, there'll be lots more. So they need to produce these plasma cells and the memory B cells, and the plasma cells will go ahead and produce the antibodies. They also stimulate the cytotoxic cells, the TC cells, into what are called active TC cells and memory T cells. It's just getting really complicated, but it's all there to help us. So these guys then, you get this proliferation, this massive, very rapid reproduction of the B cells, specific B cells, which then produce the very specific antibodies for that antigen. So it's a whole sequence of events to get rid of whatever's attacked us. But as well as that, remember we had those memory B cells. They also we have the memory T cells, and they go around finding the pathogen, and then they send out signal cytokines, and then the whole thing just keeps going. So if it's a massive infection, your body does not feel good because all this is happening And eventually everything settles down, gets rid of everything, hopefully, gets put away, everything's digested, and the body gets back to normal. And having a temperature when you're infected is also to reduce the activity of the antigens and increase the activity of these cells. So remember the T cells, we have a cell-mediated response. So cells that have been infected by a virus and which add the part of the viral protein code to their membrane, then the cytotoxic T cells recognize, remember, so if we have a cell somewhere in the body, it's infected, and if you remember, I didn't mention this time, but a virus goes in, it adds DNA or RNA to the cell system, takes over the protein producing mechanism in the cell. But the cell, even though it's being taken over, will take a section of the coat of the cell, of the virus that's attacked it, and put it on its membrane. So cells that have been infected by the virus take a bit of the protein coat of the virus, that's the antigen, on their membrane, and the TC cells, the cytotoxic T cells, that will recognize the infection cell. And don't forget, there are also cytokines have been sent out by these infected cells into the system. So these guys follow the scent and they recognize that the body has, the cell has been infected, and they also recognize that it's a body cell that's been infected because the cell still has its class 1 marker. So the TC cells then release a protein which lies the cell, it breaks the cell membrane down, and then the cells die before the viral particles have time to fully replicate. And this also happens with cancer cells. So we do develop cancers regularly, the theory goes, but this system gets rid of them. Why sometimes this cancer isn't destroyed, I don't know. And grafts, skin grafts or tissue grafts, organ donors thing, to stop this, when you have, say, a new heart, new kidney, new whatever, your immune system has to be suppressed by these special drugs, which I have no idea about what they're called, but you don't want these new organs or tissues to be killed off by your system. So the couple of diagrams. Sorry, I've got a message here. So you've got the antigen. It's engulfed by a macrophage, a large white cell. It becomes an APC, an antigen presenting cell, and to a helper cell. A B cell, this antigen can also directly activate a B cell, or the helper T cell with the antigen on its surface can get to the B cell. It goes into the plasma cells, produces antibodies. Here, the memory helper T cell stimulates a memory B cell, and then you get a secondary response. So it increases the numbers, and then on the other side, the right side, is the same sort of thing with the cytotoxic T cells, and you get the activity there, and the memory T cell, and this sequence here, the red, it increases that increases the memory cells when you get an infection. So the bigger the infection, the more memory cells are produced. And the yellow and the green are, as we said before, humoral and cell-mediated immunity. Here's another diagram. Here's a macrophage, here's a virus. The antigens on the surface of the virus are placed on the surface of the macrophage. Helper T cells recognizes these. It stimulates cytotoxic T cells, passes on the antigen, and then, which finds it and destroys it. On this side, the B cells go through the same process, produce antibodies, and help destroy it. The same with the virus. Now, I thought because COVID is very much with us, SARS, just something, the virus itself is called SARS-CoV-2 virus. SARS-CoV-1 virus was the one which was involved in the Ebola virus. Ebola. So the SARS, so that's the virus. That part is a severe acute respiratory syndrome. The CoV is coronavirus. 2 refers to the second version. And it's only the second one. The first one was 2003, which is the Ebola. And COVID-19 is the actual disease, and which was a COVID infectious disease, coronavirus infectious disease then. And here's an article about this aspect of it. I just put it in. It's reading matter, homework. Now, this is about emerging concepts and technologies in vaccine development, if you're interested in the technology of it. Now, vaccinations could also provide an enticing alternative therapy against things like cancers and substance abuse. Okay, let's look at vaccine technologies, which now there are 2 basic different ones we've been exposed to. So if you've had the Pfizer, Moderna, they're the messenger RNA one, and the other ones, AstraZeneca, the other one. Anyway, so the viral one, the first one, is where the piece of virus is attached and added to your body, and then the body is reacts to the piece of protein. So I'll go through it. So the viral vector vaccines, okay. So the vector is the thing that moves it. It relies on a small section of protein, like an antigen, which we call an antigen, and it's attached to an unrelated, non-pathogenic part of the virus, of a virus. So a non-infective virus, which does not affect us, a piece is added to that and injected into us. So this was about a 40-year-old piece of technology, and it wasn't until this technology was used for the Ebola virus. So it's an emerging technology in non-viral vaccine development. So we have virus-like particles, the VLPs, and this is where they use a platform, they call a platform. And if you remember, the University of Queensland had a problem with the platform. I'll come to that in a minute. And they produce particles that mimic the structure of the original virus, like the flu virus. So that's what we get. We get the flu virus. And we also get these nanoparticle vaccines. So these VLPs contain the genome, or sorry, they do not contain the genome. That means no DNA, no RNA. It's only the protein on the outside. So they can't replicate in the cells. There's no DNA, there's no RNA going in, only a little bit of protein. And so this is very safe, because they used to put live but attenuated, modified viral vaccines once. So these vaccines are fully activate the immune system. And so VLPs are taken up by another form of white blood cells called dendritic cells. And they are processed, and remember the antigen is put on the top, on the outside of it, and the MHC class 1 and class 2 molecules so they're presented to these class 1, class 2 molecules, and they activate the immune response. These are the adaptive ones, the level 3. AstraZeneca vaccine uses this, the adenovirus. But the adenoviruses are the ones that affect the lungs, give you a cold, that sort of thing, and a section of a spike protein was attached to the adenovirus, which has been modified so it doesn't infect us. And the University of Queensland didn't want to use an adenovirus; it used a section of the HIV virus protein and as a clamp to hold it together. And the problem was, so this is the problem was that people who were injected in the trials with this Queensland University system developed false HIV negative, false HIV test results. They didn't have HIV, but it showed that they did, because they used this little bit of protein. So that had to be discounted. So now they're working on it. And they've come up with 2 new systems, and they're undergoing trials now. Now, AstraZeneca was stopped. You can't get AstraZeneca in Australia because of these clots, but these clots were really very, very minor. And for happening in people that are under 60, in Australia, only 6 people died, and others, the doctors very quickly worked out how they weren't the normal sort of clots. And as of August last year, only 6 people. That's the latest. Yesterday, I spent quite a bit of time trying to find out how many people have died due to AstraZeneca clots. And this is the latest I could find. So they now recognize these symptoms, and they have the appropriate drugs for them. Now, I found this. I put this up as I'm interested. These thrombotic situations, the clots. There were 48. Now I put the whole lot up. This is as of June last year. I couldn't get any more information. Well, TTS says thrombocytopenia syndrome. So thrombo is in clot, cytopenia is the cell, and the syndrome is clotting thing. 48 there were reports, and 35 were confirmed, and 13 were probable. They didn't know what they were, but there may have been or may not have been. Of the 31, of these, 31 have been discharged from hospital, recovering. 15 remain in hospital. This is at June last year, including one who remains critically ill in intensive care. But so there weren't many, by August, only 6 have died. So now, what is a clot? This is important, and the clots form when they become semi-solid, when the blood clots sticks together, and that could be triggered by an injury. There could be something else in the vessels, and I should mention that we have these platelets. They're very, very important, and it's these platelets that clot. So things like aspirin reduce the sticking power of these platelets. So now there are 15 parts of the body. I did some research. I had no idea. I thought the clots, but there are 15 parts of the body that can develop clots. So DVT, so deep vein thrombosis in the legs, you get when you fly, and you get those. Remember, I mentioned the lymphatic system. Now, the blood moves around the body by the pumping of the heart, but the lymphatic system relies on our muscles moving and our guts moving, which they do all the time, to move the lymph inside the lymph veins to go. If you're sitting on a plane for hours without moving, the lymph accumulates in your legs, and then the fluid comes out of the lymph system, and your feet swell. If you take your shoes off, you may have problems putting them back on. And that's why it's very highly recommended that you move. If you go for a walk, you move your feet up and down, you paddle around, you tense your muscles, because that's how the system goes. It also happens with the blood in the system, in the legs. They need muscles to move them because there's not enough pressure from the heart to move the venous side, especially from the legs, up back to the heart. So you've got to move your legs and the muscles in your legs to get the blood. And then if the blood has been sitting there for quite a while, then the clots start to form. Another one is the pulmonary embolism that's in the lungs. Then you also get myocardial infarction, which is you get myo is muscle, so cardiac muscle clotting, and you get a heart attack where you get clots in the muscle, in the arteries that take blood to the heart. On the surface of the heart, you go into the heart, but if there's a clot there, then the heart doesn't get that. Now there's a slideshow here of these 15 different clots, and I'll leave it for you because I've just about run out of time. It's big. So on rare occasions, the clots occur, but we pretty much know how to get rid of them now. For some, it doesn't occur in the mRNA vaccines. These clots are immune system induced. So in the lungs where these clots occur, and there are also clots that go to the brain, but the ones in the lung are where the immune system misinterprets the combination of the virus and the lungs wall. So there's a big mix-up by the immune system there. and it triggers a chain of events where the platelets become activated and they stick together. Then immune system keeps activating the platelets, so this prevents the fluid moving through, the blood moving through these veins. These clots lead to a shortage of oxygen in the body, and now they know. And these are not, so we can produce clots regularly, they're not uncommon. And often the body will just get rid of them. But the TGA found that there are 50 blood clots occur daily in Australia. Normally, and there's a big uproar that there were a small number with the AstraZeneca. Now the other one is this nanoparticle mRNA vaccines. So Pfizer and Moderna are those, and they're very, very important in this with this mRNA. Now, what basically they put them into these small lipid nano spheres called mycelials, the little bubbles of fat, and they're sometimes called mycelials and sometimes liposomes. So the liposomes have a double, it's like this is a membrane. All our membranes are bilayers. So the fat's like this. If a bit of membrane becomes a liposome, so you get the inside bit, it's hollow there, and the outside. A mycel is like that. And these two can carry drugs around. And this is how they vary them. But these two carry the mRNA around. So when I should mention what the mRNA is. A piece when the body wants to produce a protein, a piece of the gene, which is a section of DNA, is transcribed. I go in detail in another talk about this. I'm going to talk about genetics, but so a transcription is the copying of the gene into RNA, and you call it messenger RNA, which then goes out of the nucleus and gets translated into the protein. Okay, so what the vaccine does, there's the piece of mRNA, messenger RNA, which is what this thing is here, is artificially made to pretend that it's carrying the virus, a section of the protein spike of the virus, and that's injected into people. The little liposome or the mycel goes into the cell. The mRNA comes out of the cell because the cell itself takes the mycel apart, and then the mRNA goes into the mechanism that produces proteins and produces multiple copies of a section of the coronavirus spike protein. And these spike proteins are released by the cell. And then the immune system recognizes them as non-self. And then everything we talked about, we get a reaction and produce antibodies, which will then be there, and the message and the memory B cells and the memory T cells are produced. Okay, so the antibodies and the memory cells are produced, and that's the whole idea. And then we get a response. Now, you all have had PCR tests, and most people know that PCR is called PCR. It stands for polymerase chain reaction. And the polymerase chain reaction is a system, it's been around for quite a while. It's about it's a small machine about the size of a small shoebox. And what it does, it can take a small piece of DNA, and it very quickly can produce millions to billions of copies of that piece of DNA. And that can be then sampled and run and identified. These days, if there's a burglary, if there's a murder, a rape, or whatever, a tiny bit of DNA left. And a guy, there was a burglary not that long ago, and the guy found a drink, a canned Coke in the fridge, so he had some of it, put on the table. The police took that can away, and there was enough DNA from his lips on the can. He was on their file. They arrested him. If there's a smoker, there are enough cells on the cigarette butt for people to identify who that is. Now, we have different sorts of immunity. We have active and passive. So active, we produce the antibodies. Passive, the antibodies come from the external of the body. And then each of these have a natural way and an induced way. So natural active immunity is when the person is infected and we produce antibodies, what we've been talking about. And then B and T memory cells are produced. That's active, natural active immunity. Now, induced or artificial active immunity is when we get immunized, when we get injections, and what we've been talking about. And the antibodies are then the primary antibody that are produced, become the primary antibody response to anything that infects us. And in this case, just remember that graph. Now the passive immunity, the natural one, gives immediate protection. So a fetus gets antibodies from the mother across the placenta, and it also gets antibodies through breastfeeding. And this is a problem for mothers who can't or refuse to breastfeed and they bottle feed, and the baby's missing out on all this. Induced passive immunity is when if you get an antitoxin injected into you, so tetanus antitoxin, or you get a snake bite, and you get injected. That's induced passive immunity. You don't actually produce it, and it's immediate. Now, I put this in. I thought you might be interested in the human HIV. So it's human immunodeficiency virus. It has got this trick, and when it infects, it often hides, and it could be hiding for a long time. And it enters the T helper cells, and it becomes invisible. It's out of the system, and it's within the cells. And it could be there for years. And once active, the T cells can't protect the body, and then you get what's called AIDS, and the people who have AIDS develop these amazingly unusual conditions. One of them is a skin cancer called it's called Kaposi's, I think. Anyway, it's quite difficult. We also have allergies. All sorts of things in our environment can affect us. Now, not all of us get them, and even if we do get them, we're probably fairly specific. So the things that cause an allergy reaction are called allergens, and it could be dust mites, pets, pollen, insects, ticks, mold, foods, and some medication. Now, atopy is a genetic tendency to develop allergic diseases. So there are people who get lots of allergies. So when atopic people are exposed to the allergens, the immune reaction leads to great inflammation. They have all sorts of problems. And so you can have nose and eyes, we can rhinitis, hay fever, conjunctivitis. These are some examples. We can get eczema, hives, asthma, of course. And when you have an allergy to an allergen, you have an allergic reaction which occurs, so like pollen, enters the body, you get antibody response. The antibodies attach themselves to the mast cells, which produce the histamines. You get the allergy, the itchy nose. Most of us would know that to some degree. Then you get inflammation, the swelling, the itchiness, the heat. And and some chemicals as well. You get adverse reactions. So they're not immune systems. You get, they're not allergies either. They're just another reaction. So parts of the body that could be affected, nose, eyes, sinuses, and so you get all this runny nose, runny eyes, itching, sneezing, watering eyes. The lungs, you get with asthma, you get everything's really sealed up. The lining of the bronchi swell, so people have lots of breathing, and you can see the diagram down here. That's normal, and you get something causing the asthma. You get the swelling. Stomach and bowel foods like peanuts, seafood, dairy, eggs. Now the peanuts are putting in Israel, kids are fed peanut butter right from when they start eating, and there's no peanut or any nut allergy in Israel. When my first grandchild was born, I said to my daughter, when you start feeding her, give her small amounts of peanut butter. She'd love it. And both kids had that. Both kids have got no issue with any sort of allergy. But my grandson is a celiac, but that's another thing. The same, you've got to feed them right from the beginning. Cow's milk allergy can occur, get all these others I'm rushing now. Some people are lactose intolerant. But it's a milk, so it's a sugar in the milk. And you get, but it's not an allergen, it's just a reaction, it's an intolerance. Celiac disease is not an allergy, it's an autoimmune disease. And if my son-in-law and my grandson who both are celiacs, have a little bit of gluten, to which they have a reaction to their villi in the small intestine disappear. So they will have a lot of trouble feeding getting any nutrients. Okay, so it's not an allergy. You can go through this again. I'm just getting skin. You get atopic things. You get dermatitis, urticaria, hives. Things that require urgent treatment are where you get anaphylactic shocks of kids who have a peanut allergy, and then somehow they eat a bit of something nut, everything swells up. They have to have their EpiPens handy. You can get anaphylactic shocks from a bee sting if you have one before. Medications, foods, and this is really nasty. Very, very rapid reaction has to happen, and so you have to. And at school, I was a first aid person for a long, long time. If kids had EpiPens, we often left them with me in case they collapsed in class or something. So So it's important to be able to prevent and avoid or minimize these things. Medications, antihistamines, all these EpiPens, of course. There are non-medicated things like nasal sprays to clear out if you have hay fever. By having the saline spray, you often can clear out the allergen. Immunotherapy, so long-term desensitization. It works for some, not others. You can get mechanical. Oh, I thought I mentioned plants. Plants have a cuticle to stop things getting in, the waxy layer. It's actually originally to stop the water evaporating out of it. But it also, it's like our skin. They have epidermal skin like us, like bark. They have chemical barriers like tobacco plants make nicotine. It's not for us, for humans to enjoy smoking. It's to kill insects that eat them. Plants also have innate passive systems. They don't have antibodies, but they have surface receptors which identify certain pathogens, and they have a mechanism, a defense system, and a lot of them have cyanide. So they direct the cyanide, they produce it towards the insects, they're attacking them, and fungi. It's fairly quick. Cyanide is in many foods, cassava, lima, beans, almonds. So the pip of the almond does have a small amount of cyanide in it. And in fact, if you can smell, if there's some cyanide gas around and you can smell almonds, say goodbye to whoever's next to you, because you're not going to live for more than a minute. So we're very close to the end of here. Okay, so I mentioned the pips. Other chemicals, latex, alkaloids like nicotine, caffeine, morphine, cocaine, they're all produced by plants to protect themselves. And these, there are also other compounds that have other mechanisms. These chemicals, there are many of them, I just listed 3 that they produce. They also have things like thorns, prickles, spines. These idioblasts are like razor sharp crystals, and which cause pain. They're often silicon. They take silicon out of the soil and make little spikes, and that's it. This is probably, I rushed it a bit near the end. It's getting late. It's anyway, so a couple of people had to go. They had messages, but there are a couple of links there which might be worth having a look at. Go back to it. It's all in the notes. I hope I didn't rush it too much, but you'll have access to it, and it's recording anyway. I'll put it up eventually. Now, I mentioned that, I only mentioned before when talking, I'm going to go, Pat and I are going next week for 2 weeks to Flinders Ranges. So today, fortnight, I think it's the 5th of May, will not be a lesson, but I'll send an email out probably before we go. I don't know if I'll be able to do it.
It's the 3rd of May.
The
3rd of May.
3rd of May, whatever it is, in the fortnight. Thank you. And I'll see you in 4 weeks.
Thank you.
Thank you.
Enjoy the rest of those of you are enjoying your matcha. And
Tell him to see out to that.
Enjoy your trip.
Ruben,
yes.
Have a good trip.
Thank you. Chag Pesach Sameach. All the best.
Ruben, can I ask a question?
All
Sorry.
the best. Helena, and
Yeah, I just want to ask a
get
question.
better.
Can I ask you a question, Ruben?
Sure.
Well, I'm very allergic to flu injection and penicillin and so on, and I'm due to get the injection now. It's very hard for me to know what to do.
You have to. It's probably not the actual flu part. It's the whatever it's being carried in. It's the liquid.
Yeah.
So with a flu shot, I think different companies make them. CSL certainly makes them. I don't know who else. You need to ask your doctor which ones you've reacted to before. He'd have a record, and maybe get another one.
All right. Yeah, they want me
And
to
the
hospital.
penicillin is often a reaction to the penicillin.
She's a scientist.
Ruben, I was
Yeah.
going to mention to you about the peanuts. I gave peanuts to my children, as you said, at 6 months.
Yes.
But I actually found out one of my children, when she became in puberty, she got allergic to walnuts and pecan nuts, which was unusual. And even the doctor thought it was unusual because I breastfed all mine and did everything the right way. But it just happens that she can have peanuts and
Yep.
peanut butter, but not those two other nuts.
Wow.
Yeah.
I hadn't heard of that. Yeah.
Yeah.
Wow.
Neither do we. My mum was even puzzled too. But yes, so but you can have anything else, but you can't have those two nuts. Mm.
Okay. Well, okay. Anyway, I need to go.
Thank you.
Guys, all the very best. And
Okay,
I'll
thank
see
you.
in
Thank
4
you.
weeks.
Bye-bye.
Bye-bye. Bye.
Bye.